All right, today we're taking a journey deep inside a microscopic world. We're going to pull back the curtain on the tiny protein factories that are humming away inside every single prokaryotic cell. This is where the blueprints of life are turned into the actual machines that do all the work. It's an incredible process, so let's get into it.
So here's our game plan. First, we'll look at the blueprint itself. Then, we'll meet the key players on the assembly team. After that, we're going to follow the whole process, act by act, through initiation, elongation, and finally, termination. Before we dive into the nitty-gritty of how it all works, let's just take a second to talk about the why. What is the point of all this? Well, at its heart, it's about turning pure information, a genetic code, into a physical, functional machine that can actually do something. And that process is called translation. You know, the name is just perfect. Think about it. If the cell's DNA is like this master library of blueprints, all locked away and protected, then an mRNA molecule is like a photocopy. It's a working copy of one specific blueprint brought out onto the factory floor, ready to be read. Okay, so you can't have a factory without a crew and some serious machinery, right? So let's meet the essential team that makes this whole protein building operation possible. First up, we've got the blueprint itself, messenger RNA, or mRNA for short. Its instructions are written in this really elegant language made up of three-letter words. We call these codons. Each and every codon is a direct command, telling the factory exactly which building block to add next. And where does all this happen? Right here, at the factory itself, the ribosome. In prokaryotes, it's got two main parts, a small 30S piece and a larger 50S piece that clamp down onto the mRNA. But here's the really wild part. The ribosome isn't just a workbench. Its own RNA, the rRNA, actually has enzymatic power. It's a ribozyme, an enzyme made of RNA, and it's what physically builds the protein. How cool is that? So how do all the building blocks, the amino acids, get to the factory floor? Well, that's the job of transfer RNA, or tRNA. These guys are basically the specialized delivery trucks. Each tRNA is built to carry one and only one specific amino acid, and it has a special tag on it, an anticodon that's like a key looking for its one matching lock on that mRNA blueprint.
And here's exactly how that beautiful lock and key system works. Let's say the mRNA blueprint reads AAG. That's the code for an amino acid called lysine. Well, only the tRNA delivery truck with the perfectly complementary anticodon, UUC, is going to fit into that slot. It recognizes the signal, docks for a moment, and drops off its cargo. It is incredibly precise.
And of course, just like any real factory, this one needs power and a support crew. You've got specialized enzymes to load up those tRNA trucks and to forge the bonds in the protein chain. You need energy, which comes from molecules like ATP and GTP. And you've got these special helper proteins called factors that basically act as shift supervisors for each stage of the process.
All right, the team is here, the supplies are ready. It is time to fire up the assembly line. This first phase, initiation, is all about getting every single piece in exactly the right spot to start building.
But before we even start, there's a crucial prep step. The delivery trucks have to be loaded up. This is called charging the tRNA. A specific enzyme grabs the right amino acid and, using energy from ATP, attaches it to its matching tRNA. This makes sure every truck leaves the depot with exactly the right cargo.
Okay, here's how the factory actually assembles itself. First, that small ribosomal subunit latches onto the mRNA at a specific start here signal. Then, the very first tRNA, a special one just for starting the process, locks onto the official start codon, AUG. And finally, the large ribosomal subunit comes in and clamps down over the top of everything. This step uses energy, and once it's done, the factory is officially open for business. Now we're at the main event, elongation. This is the real heart of the process. It's this repetitive, rhythmic cycle where the protein chain is built, one link, one amino acid at a time. To get how this works, you just need to know the ribosome's three key docking bays. There's the A site for the arrival of the next tRNA. There's the P site, which holds the tRNA with a growing polypeptide chain. And there's the E site, which is the exit for the now empty tRNAs. Just remember APE, arrival, polypeptide, exit. So this cycle is the engine of production. A brand new, correct tRNA arrives at the A site. Then, the ribosome's own enzymatic power forges a new bond, transferring the whole growing protein chain from the tRNA in the P site onto the newcomer in the A site. Then the entire ribosome scoots one codon down the mRNA. This shifts everybody over. The empty tRNA leaves from the E site, the one holding the chain moves into the P site, and the A site is now wide open for the next delivery. And it just keeps going.
And you gotta remember, this isn't magic. It takes energy to run this machine. Every single mechanical step, the arrival of a new tRNA, the ribosome shifting down the line, it's all powered by the chemical energy from a molecule called GTP.
So the protein chain gets longer and longer and longer, but how does the factory know when to stop? Well, that brings us to our final act, termination.
See, as the ribosome is just chugging along that mRNA blueprint, it eventually hits one of three special codons, UAG, UAA, or UGA. Now, these don't call for any amino acid. They're not instructions to build. They are a universal signal for stop. They are the red light at the very end of the assembly line.
So what happens when it hits that stop sign? Instead of another tRNA showing up, a special protein called a release factor slides into that open A site. This factor is like the foreman signing off on the job. It triggers the machinery to cut the finished protein chain completely free. And with the product released, the whole factory, the ribosome parts, the mRNA, everything just disassembles, ready to start a new job somewhere else.
And there it is. The final product rolls off the line, a polypeptide chain. This is the primary structure of a protein, a perfect, linear string of amino acids assembled exactly as the blueprint ordered.
But that leaves us with this one huge question, right? We've built the chain, sure, but right now it's just a long string. The real magic happens next. How does this string of amino acids fold itself into the complex three-dimensional shape that lets it actually work as an enzyme, or a motor, or a structural support? Well, that's a whole other story.